Good afternoon. These are images of some various larval habitats of members of the Anopheles gambiae species complex, particularly Anopheles gambiae sensu strictu. You can see that there's some good variety here, and our general thinking is that the ability of Anopheles gambiae to adapt to and colonize so many different larval habitats is one of the things that makes it such an effective and devastating malaria vector. One example of that adaptation is the topic of my talk, Anopheles gambiae Bamako. Bamako is a chromosomal form. It's specified by inversions 2RJ, C, and U on 2R, and it also seems to be an ecotype. It appears to prefer a particular habitat, which is these rock pools that can be found along the banks of the Niger River and its tributaries. What Bamako is not is a species. Unlike the good species in the gambiae complex, so far nobody has found any unique and identifying rDNA variants for Bamako. But given its unique chromosomal complement, its habitat preference, and its habit of assortative mating, by which I mean Bamako mosquito will generally prefer to mate with another Bamako mosquito over a non-Bamako mosquito. And by non-Bamako, I mean Anopheles gambiae sensu strictu with any other chromosomal complement. So given all those things, we were interested in this group. And I should say that our lab is certainly not the first to be interested. There's a rich Bamako literature that I encourage you to check out if you too are interested, including but not limited to these papers. But the literature doesn't always agree. So on the left, we have a figure from Nifsi et al. 2010, and on the right, we have a figure from Lee et al. 2013. And both of them are looking generally at where does differentiation between Bamako and non-Bamako accumulate in the genome. So on the left, we're looking at this top panel, and the chromosomes are arranged horizontally from left to right. And you can see that generally the highest spikes are in the inversions, particularly here in J and here in C. Whereas on the right, now we're looking down the page, down the chromosomes. If you look at this gray line, you can see that it tends to be highest on the X chromosome and in the centromeres. So findings that differentiation accumulates in the inversions would be in keeping with the theory that the inversions may be driving Bamako's adaptation to its habitat and differentiation between Bamako and non-Bamako. Whereas accumulation of differentiation on the X chromosome would be in keeping with the observation from the rest of the complex that the X chromosome is important in differentiation. So given this discrepancy, we wanted to ask 3 questions. We wanted to ask, what's the status of Bamako as an independent entity? Previous studies have suggested that it is indeed independent, but we were now in a position to address this with higher resolution data than ever before. We wanted to know, can we find candidate loci for Bamako's adaptation to its unique habitat? And can we find candidate loci for differentiation between Bamako and non-Bamako? And note that these regions and these regions don't necessarily have to be the same. To answer these questions, we used individual sampling from Western and Central Africa. We had 10 Bamako from Southern Mali, 8 non-Bamako inclusiae from Burkina Faso, and 16 non-Bamako inclusiae from Cameroon. But given the low sample sizes and the allopatric distribution, we supplemented this with pooled sequencing from Southern Mali. So this zoom is showing that most of our samples come from this one village, Kela, and we supplemented them with samples from a couple of nearby villages. From this, we made 3 pools, so Bamako, non-Bamako, and Klutsiai, and we subjected all the pools and the individuals to whole genome sequencing. So to answer our first question, is Bamako an independent entity? We used neighbor joining trees, principal component analysis, and ancestry estimation. The 3 gave roughly the same answers, so I'm going to show you the trees today. This is a tree made from SNPs across the whole genome. We have Anopheles klutzii in orange, we have non-Bamako in black, and we have Bamako in blue. The outgroup is Anopheles christii, but this branch is not to scale, and the tips are symbolized symbolizing the country that the sample comes from. So you can see that we recover Bamako as an independent group here. But there's a possible problem here. Bamako is defined by inversions 2R, J, C, and U. So if those inversions are what's driving this apparent differentiation, there could be something tautological about using this to conclude that Bamako is indeed its own group. So to address this, we also analyzed SNPs found only in collinear regions outside of the inversions. And the branch lengths are very short here, and this is in keeping with what we know of ancestral and probably ongoing gene flow among all these groups. But again, we see Bamako recovered as an independent clade. So this is suggesting that there do seem to be regions outside the inversions that are supporting Bamako as a group. So I want to call your attention to the geographic distribution here. The samples from Burkina Faso are actually a lot closer to the samples from Mali, but when we look at non-Bamako, they group with the samples from Cameroon here. So we can't rule out that isolation by distance is happening in this data set, but what we can say is that isolation by distance alone isn't enough to explain what we're seeing. Moving on to our second two questions, we used a genome scan approach to look for candidate loci. So we used 4 population genomics parameters. We have nucleotide diversity pi. We have Tajima's D, which is a measure of selection and other forces that shape the genome. We have linkage disequilibrium, or the correlation between variants. And we have FST, or differentiation between 2 populations. So we used an outlier approach in the absence of phenotypic data. And what we're really looking for are regions where all these parameters take on an extreme value. So we can see a couple of things here. Actually, I hope you can see better than I can from this angle, so if I'm pointing at something that's not quite aligned, that's why. So we see that something seems to be going on in 2RC. It looks like we might have a selective sweep within Bamako. Nucleotide diversity drops in Bamako, but not the other groups. Tajima's D really drops quite sharply. We see linkage disequilibrium elevated, and we don't think that this is due to suppressed recombination within an inversion because Bamako is defined as fixed for 2RC. So within Bamako, recombination should proceed within 2RC. And finally, FST between Bamako and non-Bamako reaches its highest value across the genome in 2RC. So this looks like a putative selective sweep, and we think something is definitely going on here. When we look at the X, we notice that, so we didn't plot data all the way out to the centromere. The assembly is a little troublesome in this region, but what we do see is the expected rise in FST between Gambi and Klutzi here, and it's not accompanied by any rise in FST between Bamako and non-Bamako in this region. We did something similar with the individuals just as confirmation, and we get similar results. We see in Bamako a drop in nucleotide diversity in 2RC. We see a drop in Tajima's D in 2RC, and a spike in FST. Here we did plot data all the way out to the centromere, and again we see the rise in FST between Gambi and Klutzi that we expect from previous research, but we see no corresponding rise in FST between Bamako and non-Bamako. This is suggesting that the X chromosome is not really implicated in differentiation between Bamako and non-Bamako. So moving on to that really interesting signal in 2RC, what's going on here? This is a view of the entire inversion, and I have highlighted where the parameters seem to take on their most extreme values. So what's in this region? You can see here there are a number of high FST SNPs between Bamako and non-Bamako, and those are circles. Sorry, synonymous high FST SNPs are circles. There are a couple of high FST non-synonymous SNPs, and those are triangles. And the black boxes are all the exons of the genes contained in the region. So you can see that there are actually 5 non-synonymous SNPs in this area. There is 1 in CYP6P3, there's 1 in CYP6P4, and there are 3 very close together in AGAP013-202. So we don't know whether these non-synonymous SNPs are driving what appears to be a selective sweep in Bamako, but we wanted to know what these genes are doing. And on that front, there's good news and there's bad news. CYP6P3 is known not only to be associated with insecticide resistance, but actually to metabolize pyrethroids directly. Similarly, CYP6P4 is associated with DDT resistance, but for AGAP 013202, we don't have any annotation. We also pulled out all the high divergence regions between Bamako and non-Bamako across the genome. We took the genes in those regions and we did functional enrichment via David to look for any themes. What we see is that the cytochrome P450s I showed on the previous slide aren't unique. There are a number of them in high divergence regions across the genome. We also seem to see some kind of signaling theme with the GPCRs here and the ligand receptor interactions here. A word of caution though, anything we conclude or theorize about the genes depends on our annotations. So it's quite possible that these putative insecticide resistance genes are doing something in Bamako that doesn't actually have anything to do with insecticide resistance, but because of our extrinsic interest in that topic, that's the annotation that we know the most about. So to conclude, in keeping with previous studies, Bamako appears to be a distinct entity on its own evolutionary trajectory. Unlike for the good species in the complex, we don't see the X chromosome implicated in this differentiation. What we do see implicated is what appears to be a selective sweep between Bamako and non-Bamako in inversion to RC. This putative selective sweep contains fixed non-synonymous SNPs in known insecticide resistance genes, but these genes are probably doing other things as well. So our approach has been able to identify a really interesting candidate region, but now we need follow-up studies to figure out what these genes are doing and to look at their phenotypic impact in Bamako. So I want to thank our funding. I also want to thank ACME for giving me a travel award to come present this research. And I want to thank some of the other people associated with this project. Due to the never predictable publication schedule, this work actually came out very recently, so you can find it here. And I thank you for your attention, and if you have any questions, I'm happy to answer them now.
Yeah, an obvious one. So what would be the history of control in pyrethroids in that region, or use of agricultural use of pyrethroids?
I know that bed nets are used in this region. The agricultural question is more interesting. It's one that I've tried to look into, and I'll admit that I've failed because the literature is in French and I'm hopelessly monolingual. So I don't know about the agricultural use. I do know that cotton is one of the main crops, and that's generally very pesticide intensive. So I would not at this time assume that an insecticide resistance sweep is necessarily associated with mosquito control, if that makes sense. Yes?
Yes, I'm just interested in the point you made that we know about these genes because they're involved in insecticide resistance, but obviously that P450s are general detoxifiers, so I'm interested about the breeding sites that you find Bamako in. Have you or anybody looked at there's anything unique about maybe xenobiotics in those breeding sites that might be driving?
That's a really interesting and long-standing hypothesis. As far as I know, nobody's looked. These are along the river, so you can easily imagine that if there is agricultural runoff in the river, it might accumulate here more than in some other breeding sites. But again, maybe that's just a just so story. We don't know yet. All right, thank you very much.
Thank you.